Von Hippel-Lindau disease is a rare tumor predisposition syndrome that leads to the formation of both benign and malignant tumors in a variety of organ systems in the body. And von Hippel-Lindau, or VHL, as I'll call it for short, is due to mutations in the von Hippel-Lindau tumor suppressor gene, or the VHL gene, on chromosome 3. And the VHL phenotype is considered to be autosomal dominant with reduced penetrance, as in the inheritance of only one mutated gene is sufficient to increase the susceptibility to tumors, such that up to 90% of individuals will have developed tumors by the age of 65. And therefore, VHL is a great example of the two-hit hypothesis, in which susceptible individuals are born with mutation in one of their alleles, but the second allele still produces a functional protein, and it's not until the second copy becomes mutated that the characteristic tumors start to develop. And so what are the characteristic tumors that develop in VHL? Well, there are several of them, and I'll try to draw them all out here to help you visualize where in the body this happens and sort of help you get a big picture, broad overview of the constellation of findings in VHL. Okay, so the first type of tumors that we'll be talking about are Cns Hemangioblastoma. And hemangioblastomas are just well-circumscribed, capillary, vessel-rich, benign neoplasms, and they're typically found in the cerebellum and in the spine. And now let's make a little bit more room before we talk about the next type of tumor. And now the next type of tumor that's commonly found are retinal hemangioblastomas, which are just hemangiomas found in the retina of the eye. Next up, we have clear cell renal carcinomas and also pheochromocytomas. And these are tumors found in the kidneys and in the adrenal glands that sit on top of the kidneys, respectively. Next up, you'll see pancreatic neuroendocrine tumors as well as pancreatic serous cystadenomas. And next up are endolymphatic sac tumors, and the endolymphatic sac is a membranous structure of the inner ear. And then the last entity we'll talk about are papillary cystadenomas, or basically cysts of either the epididymis in men or the broad ligament of the uterus in women. Alright, so these are the most common tumors that you will find in those individuals who have von Hippel-Lindau. And next up, let's just talk very briefly about the diagnosis of VHL. Make just a little bit more room to do that. So in hereditary VHL, a genetic diagnosis can be achieved with either gene sequencing or with southern blotting, which uses DNA probe hybridization. And as with all somatic mutations, there's the possibility that a patient may demonstrate what is called somatic mosaicism, meaning some cells will carry the mutation while other cells will appear to be normal. And so keeping this in mind, there's always a possibility that genetic testing may yield a false negative diagnosis.